Dr. Gayatri. Welcome you all to Bioskul. Hope you all are fine. In the previous video, we have discussed about the Kelvin cycle. In today's video, I will describe you what is photorespiration, where does photorespiration takes place, why it is called the wasteful process for the plants, and detailed mechanism of photorespiration. At the end, we will solve some previous year CSIR net question related to this topic. So guys, please watch the video till the end. So let's know what is photorespiration and its mechanism. Photorespiration, as its name, like photo means light, and respiration is the process where the oxygen is taken and carbon dioxide get released. So the process of respiration that takes place in presence of light is known as the photorespiration. But the photorespiration and the cellular respiration are different. In the cellular respiration, the ATP molecules are generated, whereas in the photorespiration process, it utilizes the ATP molecule. And in the plant, there is a process that is known as the photosynthesis. In the process of the photosynthesis, the plant, it utilizes the carbon dioxide in presence of the sunlight and it produces the carbohydrate molecule. But here in the photorespiration, what happens? The carbon dioxide, it get released. So here in the photorespiration process, the carbon dioxide which is fixed by the process of photosynthesis, it is get released in the photorespiration process. Also, it consumes the ATP. That's why the photorespiration process, it is the harmful for the plant. And this photorespiration, it is also known as the C2 cycle or the glycolate cycle. Why it is called the glycolate cycle? Because as the substrate of photorespiration is a 2-carbon compound, that is glycolate. We will see later in our slides. So, when the photorespiration takes place in plants, the photorespiration takes place when there is the bright light, high temperature, high concentration of oxygen and low concentration of carbon dioxide. We know that the plants have the stomata. Through the stomata, oxygen enter into the plant and carbon dioxide get released. Also, the plant have the transpiration process and through the stomata, water is also get released. But when the atmosphere has the high temperature, then what happened? Then the stomata, it get closed in order to prevent the loss of water. So as the stomata get closed, so what happens is the oxygen, it cannot get released out of the plant. Also the carbon dioxide, it cannot enter into the plant. Okay, so as a result, what happens? The concentration of oxygen inside the plant, it get increased, and the carbon dioxide, it is used in the process of the Kelvin cycle. So the carbon dioxide concentration also get lowered. Okay. So in that condition, when there is a concentration of oxygen is higher and the concentration of carbon dioxide get lower, then the plant, it use the photorespiration. Now we'll see where the photorespiration takes place, that is the site of photorespiration. Photorespiration, it involves the 3-cell organelles. What are those? First one is the chloroplast, then peroxisome and mitochondria. Okay. Now we'll see the mechanism of photorespiration. The enzyme, which is known as the RuBisCO, when you expand the RuBisCO, the full form is ribulose bisphosphate carboxylase oxygenates. Okay, it is the ribulose bisphosphate carboxylase oxygenates. It is the most abundant enzyme on the earth and it is a bifunctional enzyme. That is, it catalyzes the both the carboxylation reaction where carbon dioxide combines with the ribulose bisphosphate for the Kelvin cycle to initiate. But under the intense light condition, this enzyme has the ability to catalyze combination of oxygen with RuBP. That process is called the oxygenation. So the RuBisCO enzyme, it has both the carboxylase activity as well as the oxygenase activity. Okay, but that depends on the different conditions of the light and the ratio of oxygen to the carbon dioxide ratio. If the concentration of oxygen is more than that of the carbon dioxide, then the oxygen, it acts as a competitive inhibitor of carbon dioxide fixation by RuBisCO, which initiates photosynthesis. Okay. If the concentration of oxygen is more than the carbon dioxide, then the RuBisCO, it will act as a oxygenate. And the decrease in the rate of photosynthesis due to high oxygen concentration is known as the Warburg effect. So what is Warburg effect? Warburg effect is the decrease in the rate of photosynthesis due to high concentration of oxygen. Okay, this one is important. So now we will see the process of photorespiration in different cell organelles. The first, it will be started from the chloroplast. RuBP oxygenase, it oxidizes the ribulose bisphosphate and produce one molecule of phosphoglyceric acid, okay, and one molecule of phosphoglycolate. Phosphoglyceric acid, it is a 3-carbon molecule, and the phosphoglycolate, it is a 2-carbon molecule. On the first step, RuBP oxygenase, it oxidizes the ribulose bisphosphate and it produces the one molecule of 3-phosphoglyceric acid and one molecule of 2-phosphoglycolate. Then this phosphoglyceric acid, it will enter into the Kelvin cycle. I have a video on the Kelvin cycle. If you have not watched it, then please do watch that. The phosphoglyceric acid, then it will enter into the Kelvin cycle. While this phosphoglycolate, it itself cannot enter into the Kelvin cycle. And the process by which the phosphoglycolate, it converted into glyceric acid, phosphoglyceric acid, is known as the photorespiration. So, in the next step, this phosphoglycolate, it will be dephosphorylated by enzyme phosphatase into glycolate. Then, this glycolate, it will enter into the peroxisome. Okay. From chloroplast, the glycolate, it migrates to the peroxisome. So, in the peroxisome, it is oxidized into glycolate by the presence of enzyme that is known as the glycolic acid oxidase. And this glycolic acid oxidase, it is absent in the chloroplast. Okay. So, that's why the glycolate, it migrates from the chloroplast to the peroxisome, and in the peroxisome, it is oxidized into glycosylate in the presence of enzyme glycolic acid oxidase. And okay, glycolic acid in presence of glycolic acid oxidase, it forms the glycosilic acid and the hydrogen peroxide. And the hydrogen peroxide, it is removed by the enzyme catalase. In the presence of enzyme catalase, hydrogen peroxide, it is converted into water molecule. Okay. Then in the next step, this glycosilic acid, okay, in the presence of glutamate and in the presence of enzyme that is known as the glutamate glycosylate transminase, it forms glycine. Okay. So the next step, glycosylate, it is converted into glycine. So what are the steps takes place in the peroxisome? In the glycolic acid, it will be converted into, or it is oxidized into glycosylic acid. Then that glycosylic acid, it will be converted into glycine. Now the glycine which is produced in the peroxisome, that migrates into mitochondria through the amino acid translocase, which is located in the inner mitochondrial membrane. Okay, that acts as a channel through which the glycine migrates from the peroxisome into mitochondria, where the two molecules of the glycine that react to form serine with the liberation of carbon dioxide and ammonia. And this reaction, it is catalyzed by the enzyme which is known as the serine hydroxymethyl transferase. So the serine which is formed in the mitochondria, it will move back to the peroxisome. Okay, it will move back to the peroxisome from mitochondria. And in that mitochondria, it is transaminated into hydroxypyruvate. In the mitochondria, first what will happen? From the peroxisome, the glycine, it will migrated into the mitochondria. Then the glycine, it will converted into serine. Then that serine, it will move back to the peroxisome. And in the peroxisome, what happened? The serine molecule, it is transaminated into hydroxypyruvate. And for that, there is a presence of enzyme. That enzyme is known as the serine glycosylate aminotransferase. Then hydroxypyruvate, okay, in the peroxisome, it is reduced by NAD plus requiring hydroxypyruvate reductase to form the glyceric acid. Okay, so here the hydroxypyruvate, it is reduced to the glyceric acid. The glyceric acid, now it will be diffused into chloroplast from mitochondria. It will diffuse into the chloroplast, where it is phosphorylated by ATP to 3-phosphoglyceric acid in presence of enzyme glycerate kinase. Okay, so the glyceric acid, then it will be moved into the chloroplast, where it is phosphorylated in presence of ATP. So here ATP is consumed to form the 3-phosphoglyceric acid, and now this 3-phosphoglyceric acid, it will enter into the Kelvin cycle. Let's summarize the mechanism of photorespiration. If there is a high concentration of the oxygen, then RuBP, it will be act as a RuBP oxygenase. Then it converts the ribulose bisphosphate into one molecule of phosphoglyceric acid and one molecule of phosphoglycolate. Phosphoglyceric acid, it will enter into the Kelvin cycle, but the phosphoglycolate, it itself cannot enter into the Kelvin cycle. So in the photorespiration, the mechanism through which the phosphoglycolate, it get converted into the phosphoglyceric acid, then it will enter into the Kelvin cycle. So let's see what happens. In the first step, photorespiration process, it takes place through the 3 cell organelles, that is the chloroplast and the mitochondria. Both are the double membrane bound organelle, whereas the peroxisome, it is a single membrane bound cell organelle. The phosphoglycolate, which is formed in chloroplast, it will be converted into the glycolate by the enzyme phosphatase. Then this glycolate from the chloroplast, it will be moved into the peroxisome. In the peroxisome, it will be oxidized into the glycosylate, and it also produces the hydrogen peroxide. Hydrogen peroxide, by the action of the catalase which is present in the peroxisome, it converted into the water molecule. Then the glycosylate, it forms the glycine. Then the glycine, it will be moved into the mitochondria, where the glycine, it will form the serine molecule with the release of the carbon dioxide and ammonia. And the serine from the mitochondria, it will move back to the peroxisome. In the peroxisome, the serine, it will be converted into the hydroxypyruvate. Then in the hydroxypyruvate, it will converted into the glycerate. And now the glycerate from the peroxisome, it will be moved to the chloroplast, and in the chloroplast, the glycerate, it will be phosphorylated in presence of the ATP into phosphoglycerate, and now the phosphoglycerate, it will be entered into the Kelvin cycle. So you have to memorize this cycle, because mostly you will get the question like which metabolite moves from the peroxisome to the chloroplast, which metabolites it moves from that peroxisome to the mitochondria, like this. Okay, so let's see the previous year questions. During the operation of C2 oxidative photosynthetic cycle, which of the following metabolite is transported from chloroplast to peroxisome? Okay, chloroplast to peroxisome. Which one? That is the glycolate. Okay, so the answer is option D. Ribulose bisphosphate carboxylase catalyze both carboxylation and oxygenation of ribulose 1,5 bisphosphate. The latter reaction that is the oxygenation reaction, initiate a physiological process known as the photorespiration. The following are certain statements on photorespiration. Let's see the options. The active site on RuBisCO for carboxylation and oxygenation are different? No. One of the steps in photorespiration is conversion of glycine to serine. Yes, this one is correct. 50% of carbon lost in chloroplast due to oxygenation is recovered through photorespiration. No. The pathway of photorespiration involves chloroplast, peroxisome and mitochondria. This one is right. So, what is the question? Which of the following combination of above statement is correct? That is option Q and S. This option C is the correct answer. Okay. The next question. Which one of the following metabolite that moves from the mitochondria to peroxisome? Okay. During the operation of C2 oxidative photosynthetic cycle. Okay. So which one? In the mitochondria, the glycine is converted into the serine. Then the serine, it moved back from the mitochondria to peroxisome. So option D is the correct answer. Following are certain statements regarding RuBisCO. The predominant protein in plant leaves that catalyze the initial reaction of Kelvin-Benson cycle. Okay. Option the P. During oxygenase activity of RuBisCO, oxygen is used as a substrate to produce the 3-molecule compound, that is 3-phosphoglycerate and 2-carbon molecule 2-phosphoglycolate. Yes, it is produced during the oxygenase activity as we have seen in the cycle. In red and brown algae, the large subunit of RuBisCO is localized in chloroplast while small subunit is localized in the nucleus. No. The bound sugar phosphate in RuBisCO is specially removed by an ATP dependent enzyme, that is RuBisCO activase. Yes. The active form of RuBisCO catalyzes carboxylation and oxygenation reaction in 5 steps. Yes. So which of the following combination are right? That is option P, R and S. That is option D is the correct answer. Thank you so much for watching my video. If you are new to my channel and if you have not subscribed, then please do subscribe and press the bell icon so you will get notified when I will upload new videos. You know, photorespiration process is a wasteful process. So to avoid the photorespiration, some plants have adopted the C4 metabolism. So my upcoming video, it will be on the C4 cycle. So guys, please stay tuned.